Great pick! Let's talk about nucleic acids. Nucleic acids have one main function, and that is to carry information inside cells. Think of them like sets of cellular directions. Nucleic acids do not really do anything, but other things can read them and then follow the directions that were read. There are two main varieties of nucleic acids. We have ribonucleic acids, also called RNA, and we have deoxyribonucleic acids, also called DNA. So now let's dive into the structure of nucleic acids. Nucleic acid monomers, as we all know, are called nucleotides. But all nucleotides are made up of three components, which are a phosphate group, a 5-carbon sugar, and a nitrogenous base sticking out the end. Now, this here is what is the same in all nucleotides, but there are a few ways to make this configuration slightly different, which differentiates RNA from DNA in terms of their structure. The first is what's coming off of this bottom carbon here. In RNA, we have a hydroxyl group coming off that carbon, or OH. When the carbon, this 5-carbon sugar here, looks like this with a hydroxyl group, it's called ribose. Remembering that os means sugar, so it's a ribose sugar. And this is why RNA is called ribonucleic acid, because it uses ribose as one of its main components. Now, in contrast, DNA does not have this oxygen coming off the bottom carbon, so when the ribose sugar is lacking this oxygen, it's actually called deoxyribose. D meaning no, and oxy meaning oxygen. So this makes sense. DNA is called deoxyribonucleic acid, and it's because it's a nucleic acid with a ribose sugar that's lacking oxygen, so it's deoxyribo. Now let's look up here at the nitrogenous bases. I'm going to write them out really fast. Okay, so the nitrogenous bases up here, the three that I've written down for you, adenine, cytosine, and guanine, are what are shared between DNA and RNA. These are the three nitrogenous bases you can find in either DNA or RNA. It's found in both. However, there are two nitrogenous bases that are unique. One's unique to RNA and the other to DNA. So RNA's special nitrogenous base is uracil. You will not find uracil in DNA. And conversely, DNA's special nitrogenous base is thymine, and you will not find thymine in RNA. These nitrogenous bases are basically the information or directions that other molecules in the cell read or use. The order that these nitrogenous bases appear along a DNA or RNA sequence, though, is very important and very specific. Think of it kind of like code in a computer, but instead of ones and zeros, we have A's, C's, G's, U's, and T's. Now, to help us discuss the orientation of a nucleotide, scientists arbitrarily numbered off the carbons in the 5-carbon sugar. So what they did, and what we always will do, is start with the carbon that's closest to the nitrogenous base, so this one here. And then they numbered them off clockwise, like so. And then ending with 5. This is important to know because we often talk about in which direction DNA or RNA is being read or replicated, and we use these numbers to communicate this direction. Namely, we find ourselves referring to the 5' and 3' ends most frequently. This is because of how we form nucleic acid polymers. Remember, what we are looking at right now is a single nucleotide, which is a monomer of a nucleic acid. This means we are looking at just one subunit of DNA or RNA. A full strand of DNA or RNA is a polymer made up of many nucleotides bound together, where phosphodiester bonds, which is a type of covalent bond, connect the sugar of one nucleotide to the phosphate group of the next nucleotide, and so on, like so. We call this chain of phosphate groups bound to sugars the sugar-phosphate backbone, because it's the backbone of the nucleic acid polymer, and it is made up of sugars and phosphates bound together. And as you can see, when we look at this nucleic acid, at the top we see the 5' carbon, and at the bottom we see the 3' carbon. And this is the direction in which we read DNA and RNA, from 5' to 3'. Now there is one more major structural difference between RNA and DNA. We already touched on how the sugar is slightly different between RNA versus DNA, where RNA has the ribose sugar subunits and DNA has deoxyribose. And we also talked about how the nitrogenous bases for RNA are adenine, cytosine, guanine, and uracil, while for DNA they are adenine, guanine, cytosine, and thymine. But the last big structural difference is that RNA is usually single-stranded, meaning it is basically what you see on the screen right now, with just one line of nucleotides linked together. In contrast, though, DNA is usually double-stranded, meaning that there are usually 2 lines of nucleotides that run in opposite directions of one another. And these 2 polymers are bound to one another through their nitrogenous bases. The nitrogenous bases in one nucleic acid form hydrogen bonds with the nitrogenous bases in the other nucleic acid, holding the 2 strands together. The other thing that is special about DNA is that the 2 polynucleotide chains that make up the DNA strand actually wrap around themselves and form a double helix structure, like what I just drew, where these lines here represent our sugar-phosphate backbone for each polynucleotide, and coming off of each sugar-phosphate backbone are our nitrogenous bases, which are bound to the nitrogenous bases of the other strand via hydrogen bonds. Now, as it turns out, not just any nitrogenous base can bind with any other nitrogenous base. Only certain bases can bind with certain bases. So, for instance, adenine can only bind with thymine, and vice versa, and cytosine can only bond with guanine, and again vice versa. So guanine can only go with cytosine, thymine can only go with adenine. And we call these winning combinations, adenine to thymine, cytosine to guanine, base pairs, because they are pairs of nitrogenous bases bonded together. So that's an overview of the structure of nucleic acids. So before we do a deep dive into the relationships between RNA and DNA, as well as the functions of RNA and DNA, let's have a brief check-in. Check-in number 16. Take careful note that this is number 16, so you should be filling out the response to number 16 on Canvas for this question. What is the main function of nucleic acids? Feel free to pause before moving on. Number 17. Is DNA capable of doing anything? Why or why not? Number 18. There is a strand of RNA that must become bound to DNA. via hydrogen bonds between the nitrogenous bases. An image of the DNA strand that the RNA strand must bind to is shown to the right, and an incomplete RNA strand is also shown to the right. Using deductive reasoning, try to determine the nitrogenous base that belongs in the blank of the RNA strand. Note that the blank is in the RNA strand, not the DNA strand. Check-in number 19 and check-in number 20. This is a two-part question. What is the sugar phosphate backbone made up of in DNA? Be sure to include the correct name of the sugar and the bonds formed in your response. How does this compare to RNA? Check-in number 21. In regard to only what you have learned in this class so far, so making sure we're not using our previous knowledge from other classes, and we're also not using information from online, but based solely on what we have learned in this class thus far. What is the relationship between carbohydrates and nucleic acids? If you want to flag this one and come back to it after reviewing carbohydrates, that's totally fine. Okay, so we've finished the overview of the structure of RNA versus DNA. So now let's discuss their function, how they are similar and how they are different.